Okay, let's jump right in. Today, we're going to break down something that's been a complete game changer in neuroscience, the fifth edition of the World Health Organization's classification for brain and spinal cord tumors. And trust me, this isn't just some minor update, it's a whole new blueprint for how we understand these diseases.
So, let's start with a number, 22. Back in 2021, that's about how many new, officially recognized tumor types just appeared on the scene. And that makes you wonder, right? Why the sudden, massive expansion? Why did the entire map of brain tumors need to be redrawn so drastically?
Well, the answer really begins with the limits of our old tools. For over 100 years, the microscope was king. Pathologists would look at a tumor's shape, its cells, its structure, what we call its histology, and that's how they'd make a diagnosis.
But here was the huge problem. You could have two tumors that looked like identical twins under the microscope. But in one patient, that tumor would be pretty slow growing, while in another, it would be incredibly aggressive. The microscope could only tell us part of the story. It was obvious there was some deeper, invisible biology going on.
And that is where this new molecular blueprint comes in. The WHO CNS5, as it's called, marks this fundamental shift from classifying tumors by what they look like to what they truly are at a genetic level. It's a move from just appearance to their actual identity.
At the very heart of this new blueprint is this idea of an integrated diagnosis. This isn't about looking at just one thing anymore. It's a multi-layered approach, a fusion of data. You can think of it like building a complete, detailed profile of the tumor. So this integrated approach really stands on three pillars. We still start with histology, what the tumor looks like. Then we add another layer, immunohistochemistry, which tells us what proteins it's making. But the real revolution is that final step. We actually decode its molecular genetics. And it's the combination of all three that gives us a diagnosis that is incredibly precise.
So what does this actually look like in practice? Let's take a little tour of some of these new and redefined tumors to see how this whole molecular blueprint really works. A perfect example is in pediatric brain tumors. For a long time, we've known that a glioma in a kid is just a fundamentally different disease from a glioma in an adult, even if they look similar. The new classification finally puts that front and center, creating completely new categories that are based on the unique genetic mutations that drive these childhood cancers.
Now, what's really fascinating here is the naming itself. I mean, look at these names. H3K27 altered, H3G34 mutant. The specific genetic mutation is now literally part of the tumor's official name. That's not just a label. It's a precise biological address that tells us exactly what's making that tumor tick.
And the changes aren't just about adding new tumors. It's also about reclassifying old ones to better reflect their biology. For decades, we called them pituitary adenomas. Now, they're called pituitary neuroendocrine tumors, or PITNETs for short. And that name change is a big deal. It signals that we now understand these are part of a wider family of neuroendocrine tumors, and that has huge implications for research and how we treat them.
Okay, now let's look at ependymomas. This entire family of tumors is now broken down based on molecular features that are tightly linked to where they are in the brain or spine. So a supratentorial ependymoma with a ZFTA fusion, which is where two genes have mistakenly stuck together, is now understood to be a totally different disease, with a different prognosis than one with a YAP1 fusion. This is precision medicine in action, right here.
This brings us to probably the most practical part of this whole thing. How does a pathologist actually use this new map? Because it's not just a list of names. It's a logical pathway, a new way of working.
Today, pathologists work through a diagnostic algorithm. It's a step-by-step process. They start with the microscope, which gives them a ballpark idea, and then they follow this kind of decision tree of molecular tests. They're basically asking, does it have this mutation? Yes or no, to navigate their way to the most precise and accurate diagnosis possible.
And this table just illustrates that workflow perfectly for the most common adult brain tumors. So let's say a pathologist sees a diffuse glioma. The first question is, does it have an IDH mutation? If the answer is yes, they go down one path. The next question is, does it have a 1p/19q codeletion, where parts of two chromosomes are missing? If that's a yes, boom, you have an oligodendroglioma. If it's a no, you check another protein, ATRX. If that's lost, you've got an astrocytoma. This algorithm is absolutely critical for getting the diagnosis right every single time.
So why does all this molecular detail matter so much? Well, this slide says it all. In the back of the brain, the posterior fossa, we have two main molecular groups of ependymoma, PFA and PFB. PFA tumors mostly show up in kids and unfortunately have a much poorer prognosis.
PFB tumors are more common in adults and have a better outcome. They might look similar, but their genetic code tells a completely different story about that patient's future.
So you see, this isn't just some academic exercise for pathologists. This new, high-resolution map has really profound implications for every single person diagnosed with a brain tumor.
The key takeaway really is this. The final pathology report is no longer just a single observation. It is an integrated diagnosis. It's the difference between having a vague, hand-drawn sketch of a country and having a super high-resolution satellite map with layers for roads and buildings and terrain. It's just a more accurate, more powerful guide to a very complex landscape.
And that leaves us with a really critical question looking to the future. We can now define these tumors by their precise genetic drivers. The map has been drawn. The next great challenge, and really the ultimate promise of this whole new system, is developing treatments that can target those exact molecular addresses. How quickly can our therapies catch up to our diagnostics?